I have the pleasure of, I think, introducing not only a friend, but to me, friend to many of colleagues already. I think Professor Chih-Ming has been visiting with us multiple times. And we appreciate his coming back periodically to share his news finding and also to collaborate. I think Carl is here. Carl and Yi-Kun have been collaborating with Chih-Ming on some of the 当归补血汤, right? But I'd like to say a few words about Chih-Ming because I've known him probably longer than, I guess, anybody in this room. Okay, anybody else. And I've known Chih-Ming about 30 years ago. Okay. And at that time, he was still a car-carrying aerospace engineer. Since then, I think he realized aerospace is too hard, so he decided to do something simpler, easier. Okay, he has been doing very well. Okay. But I'd just like to say that in his earlier stage of career, he was very well known for his work in turbulence and some flow control related areas. In fact, some of the papers he wrote at that time, in the late 70s, early 80s, are still widely cited and followed today. Then later he switched. He started to get involved very actively in microelectronic mechanics, MEMS devices. In fact, as many people consider Chih-Ming to be one of the pioneers and the most productive researcher in MEMS areas, and many of his former students are now leading researchers and practitioners worldwide. Some of them sitting here, okay, including Yi-Kun. Some of them are in the university in the US, Taiwan, mainland China, Hong Kong, elsewhere. Okay. And in the last 10 years or so, maybe longer, he has switched again, okay, to get involved in particular in areas related to medicine, especially in personal medicine. Okay, and in case you didn't know, this latest issue of Science Translational Medicine, the cover article was co-authored by Chih-Ming and his team, including his own son, okay, who is also a professor in UCLA. I think his son is probably, there's danger his son is going to become more famous pretty soon. Okay.
Already.
That's what every father would like to see, okay? But today I think he's going to share with us some of the, I think, thinking and the experience and the latest finding with us. And I think we're in for a treat. Let's welcome our speaker.
Okay, so we are talking about the personalized medicine. So personal medicine in Chinese really is just 因人而施药。因为现在 when we want to sick and to get to see a doctor, and they give you the same drug, same dose to the same disease. Okay, but actually that's most time is not really very effective. And so now we try to find a way, try to be able to give the, depends on the person, depends on individual specific patient, and we can give the right drug and the right dose to that person. And what we do is it's very different from the, most of the people trying to do is from genome side. We try to do from the phenotype side, so from output, from the endpoint of the patient response to the drug. And so before we start going to the personalized medicine, let's look into the pharmaceutical practice nowadays, especially in the pharmaceutical industry. So if we talk about the monotherapy, that means we treat the patient with one drug. Okay? And some of that is treat some disease we treat with one drug. If you want to develop a new molecule for treat a patient, and usually development costs about $4 to $8 billion for develop a new drug molecule, and takes 10 to 15 years. So after all the spending in the time and also in the money, and then when we start to use this drug, and not that difficult, because we know that if we increase the efficacy and we increase the dosage, we will have high efficacy, and also at the same time also high toxicity. So that's pretty easy to work on that. And then on the other hand, most of the disease and has to use combinatory drug, especially the disease which mutate very fast, and we do need to use different drugs to try to treat this problem. And when we use different drugs and use combinatory drugs, and the problem really becomes very, very difficult, because high dose does not mean high efficacy, and due to the drug and the drug interaction. And that makes our selection of the drug and the dose become exponential increase. So if we have M drug with N dose, and we'll take N to the nth possible combination, and how we are going to be able to find out the best drug and the dose for the patient, and especially for specific patient. Okay? And another thing, what during our study, and sometimes we start to search for the drug combination, and we put a few drugs together, and we have a large number of different drug combination. And most time, and only a very small number of the combination has high efficacy. And the majority of the combination doesn't have efficacy. And by doing that, so if we do try and error, like nowadays what we're doing, and most likely it will not be successful. So when we talk about the challenge of the current way for the drug development, and in cell line test, and because there's a very large number, n to the nth power of the drug dose search space, and we just try a few of that and find a good one. The good one doesn't mean it's optimal, and most times it's suboptimal. If you're based on suboptimal, and then you go from cell line to preclinical test, and from preclinical test to the clinical test, and the chance become less and less to be successful. And end up with successful rate for drug development, and from the cell line until to FDA approval is only a few %. One drug takes so much money, so much time, is because the successful rate is very, very low. Okay? And so this is the challenge what we're facing, and that's the way we're doing business now. So we do need to think differently, and we cannot continue by doing that. And the few years ago, we talked about is $1 to $2 billion. Now we're talking about $4 to $8 billion, because the number of drug molecules become less, and then a lot of things, new diseases come out, and that's all the troubles we are facing. So what we're talking about combinatory drug. Nowadays, almost all disease treated by a combinatory drug. And the cancer sometimes treated by targeted drugs because we do not know how to use targeted drugs to do combinatory therapy. If we know, and we definitely will use the combinatory targeted drug. Let's take one example. What we work with a group at Switzerland and Belgium. So what so this is the how many days we treat the animal. And this is the size of the tumor volume. And the solid line is the control. Control, that means we do not give the drug for the group of mice. And then we use 3 different drugs with different dosage, and we treat that treated group. And it really has no effect at all. This drug doesn't work at all. Okay? But however, if we do a right combination by using proper search, and we can find that these 3 drugs, and the 2 means low dosage, high dosage. So 3 drugs and with 2 low dosage drugs, and then the efficacy becomes very good compared with the control group. So that shows if we use combinatory drug, and has a great advantage if they work synergetically, and we can have very good combination. But a good combination like that doesn't happen in all the combinations. And if you just by simple math, and a small number of drugs can come up with a very large number of combination, and only a small fraction of that has a good combination. That's why, and in the conventional approach, and the successful rate is very, very low. The most famous drug treatment by using combinatory drug is HIV. Okay? HIV was found out is really a horrible disease at the early about 1980s. And the death rate keep increasing. And even at that time, there's many drugs coming up and try to treat the patient until 1995. Reached 1995, there's a 3 drug combination and become available. Use that 3 drug combination in 2 years, the death rate dropped 60%, 60%. And from then on, the death rate keep a fairly low level and until today. Okay? The reason for that is because HIV virus and mutate frequently. If you mutate frequently, and if you don't use a combinatory drug, and then it will not be able to stop the spreading or growth of this viral load. So combinatory drug until today is still not optimized. What do we mean not optimized? Usually we know use the 3 drugs will be effective, but we do not know how to handle the dosage. Okay? David Ho, he's the one of the first guy to use this drug combination. And when we started working on HIV drug treatment, I called him up. I said, hey, how do you optimize? He said, no, we don't know how to optimize. The way we do that is called the sum of the maximum tolerated dosage. What is the maximum tolerated dosage? You take in the phase 1 test, you take healthy people, you give in the drug, 1 drug, and increase the dose until the patient cannot stand, not patient, until the healthy volunteer cannot stand, you take half of them. Okay? Then you add another drug. Increase the dose, and then to the maximum, they take half of them. So you take 3 of the drug until reach the maximum tolerated dose, and then you try to treat the patient. And this kind of way of thinking is linear. Okay? You add 3 of them, put it together, it will work. And never cannot take the drug-drug interactions. Okay? And so you have to try many of that until you find 1 drug and a dose combination and it works, then just continue to use that. Okay, so combinatory drug definitely is very powerful. And then the problem is how do we come out with a really optimized drug combination? And so first thing, you have to discover a target. Okay? In such a complex protein network, and you have to find out 1 protein doesn't work right, 1 pathway doesn't work right. Okay, so that's really is find a needle in haystack. And then you have find a drug can conjugate on this diseased on this protein, and then make try to either kill the cell, or you can get the cell back to healthy state. And so that's also a very tough project, right? Because you have to find another molecule can conjugate and then do the right job. And usually 1 target doesn't work, like HIV and cancers, many of them. So you have to find many targets. After you find many targets, you have to find many drugs. When you find many drugs, and then you have to be able to, this drug can work synergetically and finally can treat the patient. And then the problem is not only the mechanism or the target drug is important, and the dosage matters a lot. Okay? So you have to get the dosage right. So and the dosage right, then the possible combination is exponential number. So okay, we spend all the money, spend all the time, and spend all the effort, and we get the right drug or drug combination. And we start to treat the patient. So that's how the result when you treat the patient. That's for cancer. Okay, the cancer response rate, and the breast cancer, colon cancer, etc., and it's about 20 to 50% or 30%. When you go to liver cancer, etc., it's about 5 to 15%. When we started working on this project, and one of the very top the lung cancer specialist at the UCLA, Steve Dubinack. He came to my office, and when he sit down, and the first few sentences, he said, you know how much I can do for the patient? A patient comes to my office and give the drug and try to treat the patient, and only 15% of the patient has a very good response. 50% of the patient has no response, and the 35 is from very good to 0. Okay, so that's fall into that range. So why is that? The reason for that is when we talk about the lung cancer is not one type of lung cancer. It's usually about 20, 30 different lung cancers. And then the subtypes, for each patient, the subtypes are not all the same. okay, and the mixture is not all the same. So because there's a large cancer heterogeneities, and also human are different, okay? And black people different from white, white different from yellow, and even for us, we are all from the same race, and each patient will respond to that differently. So the human diversity and the cancer heterogeneity, and is really the main reason for this very low cure rate. Okay, and that's not only for cancer. Cancer has a fairly low cure rate. Some of it has high cure rate, but still there are very different response. So we do need to go to personalized medicine. So we go to the personalized medicine. There are many different ways to do that. One of the most common way is from a genotypic approach. So from genome, and then produce all these proteins. Then proteins, they form pathways. Pathways are all intact. So it's really a very, very complex network in the protein level. Okay? And then we have to find out the target we have to treat, and then we have to hope we'll be able to be successful. Okay? So this is a reductionist hypothesis-based approach. So we think in this cell, and which one of the protein or pathway has problems, okay, which is caused from the genome and go to produce a protein. The protein does not function right, and then we try to find drugs and treat, and then we hope the whole body, the patient would be able to be treated and become healthy again. Okay? However, this approach is not a one-to-one relationship, and also the distance between genome to the phenotype is extremely far. The reason for that, first, the genome is different for each person. Okay, there's no such called identical twin have the identical genome. We did at the very beginning when the research, we did one experiment. We take one single cell. We take one single cell, let it divide 10 times. After divide 10 times, has 1,024 cells. Okay, then we put the same drug to treat these 1,024 cells, and the response split into two groups already from one single cell. Okay? And there's a lot of fundamental study in biology why one cell divided into these two cells already have different genotypic type of construction already. So epigenetic effect, and that's for different people, and a lot of disease mostly is caused by epigenetic effect rather than by genome effect. And from the genome to the protein is not a one-to-one relationship. And so if we wanted to, based on genome, go to the phenotype, the correlation is very weak. Okay? And there are things are pretty, you can find that the common features for people who have a similar genome profile, but most of them are not. That's reflected by the cancer response rate. Okay? And so when we start to talk about from genome to protein network and the phenotype, we're really talking about very, very big data. Big data is a popular buzzword nowadays. Okay? So when we treat the patient, it's not only this big data, there's another big data. Okay? And they have to take combinatory drug, they have to take a lot of drugs. So when these people take these drugs, this drug will interact, each of the drug molecule will interact with some part of the pathway, and so many drugs is going to interact with a complex protein work, and then it's not just too big data set. Okay? When they put it all together, it's infinite data space. So it's really almost a mission impossible to try to go from the genotype and try to treat a specific patient. And when you see this picture, some people laugh. And sometimes I thought, yeah, that's a picture we got from Google. Maybe that's a picture just illustrate how much drug patient will use. And actually, when we started working on the first personalized medicine, which is liver transplant, okay? And first, the doctor said, okay, we use 3 drugs to control the one of the marker. And so we said that would be very simple. Then we start to work on that. And work on it pretty good. And a few days later, and certainly it doesn't work. And then we called the doctor, what's going on? And the doctor, oh yeah, seems that patient was infected by something because it's immunosuppression. So in fact, so we give the we give antibiotics. They say, hey, when you give antibiotic, you have to let us know because our drug, our whole method is based on input. Then we went to his office and then said, tell me what all the drugs you use. He gave me a long list, like that long. And then he said, yeah, this picture is real. The patient take that many drugs. Okay? Because you have to take the antibiotics, you have to take all the immunosuppression, and then you have to take vitamins, you have to take many, many things. Okay? So then we start to realize a very large big data space intact with another one, and which become almost infinite data space. So if you want to do a personalized, and it's a mission impossible if we take that way. So when we reach that stage, we have to think very, very differently. Okay? Just like Alexander the Great, he wanted to go the Middle East, and he wanted to untie the Gordian knot. He cannot do it just like everybody else cannot do it. So he takes his sword and cut it open. Okay? So we have to use a very different approach to solve a problem which we think is impossible, use the traditional way. So we took an orthodox approach. So let's bypass the big data. Yes, big data has a very good theoretical and all the knowledge base if we want to go from genome to that way, but we know that it will be very difficult for us to do that. So let's bypass the big data. Don't try to write a proposal to NIH asking for big data proposals. Money, okay? Okay, so what do? We take an orthodox top-down system approach. I'm an engineer. Okay, so we always think it's a system. We always think from the top down rather than we go from the bottom up. Okay, so what do? We really look at the output, the desired, what do we want to, the desired phenotypic output. Okay, and so we say that's what we want to cure the patient, and then that's our desired output. Then we find out how this drug dose input. The drug dose input will interact with the DNA, protein, and the cells, organs, or even the animals and to the body until we'll be able to successful. So what we are thinking is just look at input and output, whether we can find a simple way. And we can relate the input to output, and then after that, if what is the best output we want to have, then I can tell the doctor to use what kind of best drug and dose combination. Okay? And so that's what we intend to do, but we really don't know whether we'll be successful in view of all this such complex structure. The first experiment, which this result I have presented last time when I gave a talk here maybe 3 years ago. So at that time, we used engineering feedback control concept. That's why I call the FSC technique, and FSC-I is generation 1 technology. So we look into how does HSV-1 viral infection infect the 3T3 fibroblast. So our friends in biology try to help to code the virus, and if the virus ever get into the cell, then have green fluorescent protein expressed. So we see the green fluorescent light. Then we pick up 6 drugs. Each one has 10 dosage. We have 1 million possibilities. We start with arbitrary one and put it into the Petri dish, measure how much green fluorescent light we observed. That means how many % of the cell has been infected. And we wanted to have a non-infected cell be 100% non-infected cell. That's our desired output. Of course, the first one is arbitrary choice, so will not be very good. So then we use search algorithm help us try to going around and try to improve the input until we get the optimal output. From 1 million possible combination, to our surprise, it takes about 10 to 20 iterations. We find out 0% infection. Okay? And another thing very interesting we have found out, these 3 drugs have 100% inhibition. This is a log scale. And if we change this one by 2 orders of magnitude, that's the main drug to treat the HSV-1 viral infection. And also we increase the dosage by about a factor of 2. And actually 20% get infected. So high dose doesn't mean high efficacy. Okay? So the dose is an important part to try to making the treatment correct. And that means the search space becomes exponential number. Okay, so that's how we needed to find a way, a simple way, try to optimize the drug combination. So we did that. We did it on many indications, many kind of disease works also all very well. Just 10 to 15 iterations, we can reduce from 1 million, 10 million possibilities down to right point. We want to do that. And the reason we can do that is because we can kill millions of cells. Okay? We can have that luxury to do that, and it can do it right. Then we did estimate if we want to do this feedback control for animal, for mice, we have to kill about 2,000 animals in order to get a right optimal value. Of course, that's not allowed. And so we stuck there for quite a while. Okay, so how do we go from the feedback control to be able to use a sacrifice small number of animal, and we can get the optimal drug dose combination. And when we go to human, we cannot sacrifice single human. Okay? So that's the real challenge there. After we find out that we can do it with a small number of iterations. So if that's the case, we are still talking about several order of magnitude improvement. So we cannot just follow the same way to do that and just try by trial and error. So we really want to see, can we find the law of nature for biosystem? If we can, then we can use that law to apply to all the indications, all the biosystems, cell, animal, and human. Okay? And then we'll really be able to achieve what we want to do. That's a big challenge, and especially due to human diversity, epigenetic effect, cancer heterogeneity, and also the drug dose combination. For such a large number of possible effects will affect to diseased people. And so we have to, we hope, can find a simple law of nature for the biosystem. And one of the reasons to do that, in addition, we cannot use too many of the test subjects to do the test. And when we start to treat the patient, the most critical limit is the time limitation. You cannot try this, try that, and etc. Because when you treat the patient, you have very short time duration you can treat. Okay? And for example, like an infectious disease, like Ebola, you have 2 weeks. If you cannot do that, anything, the patient will die. You cannot do many trials and do this. You have to do it very fast, and you can get it right. So this is the most critical limit for when you start treating patients. Okay? Now we can treat the single patient specific very fast, and we still have faced a lot of problems when we go to more challenge. That's why I work with Wang Yang, try to find a way can we further reduce the number of tests and can do it can get the right drug dose, right time. So let's take the first law we learned. Okay, F equal to MA. So if I put F, that's my input, okay, and this output is A. Okay, so how do I relate F equal to MA? That's famous law, right? F equal to MA. This input is output that is a constant. So mass is a calibration constant in this case. Okay? So can we find something so simple in the biosystem? And the biosystem is so complex. And if we can find that, then there's hope we can really be able to treat a specific patient. So after a lot of tests and etc., and then we go to FSC.2, that's second generation. Actually, we start to try to move from FSC to PRS, parabolic response surface. That's one of our first experiments, the herpesvirus infection. So my students start to plot all this response curve. Vertical axis is the efficacy. 2 horizontal axes are the drug dosage. So they plot that, and we discuss in the meetings or in the progress discussions, and it's a very smooth surface. Then for 6 drugs, and you can plot the 20 of this surface. Everyone is a very smooth surface. Then there's non-small cell lung cancer. You plot the efficacy drug dose, is a very smooth surface. After we see so many smooth surface, I say, hey, what type of smooth surface is that? And find out it's second-order algebraic function. Okay, constant first-order, second-order drug-drug interaction. So all that we have that time, we have done about 10 different diseases already. When you plot all that, it's a second-order system. It's algebraic, second-order algebraic. So it's a parabolic surface. So we plot all of that, and you see all the case for the liver transplant, the paper we just published, and every one of that is a different type of parabolic surface. Okay? So this kind of parabolic response surface really represents a systematic patient and the response to the drug. And so that's the key. That's the law of nature for a personalized medicine. We use that simple surface and we'll be able to really treat a single patient. So next two slides is the focal point, and it's the key of the phenotypic personalized medicine. So it's a third generation of FSC technology. S prime is a mechanism causing the disease, S prime. And it takes us a lot of effort in order to find out of that. Then the patient getting sick because of the S prime, and so that's a function of phenotype, function of a diseased cell or diseased body. And we don't know how to write this equation. Okay? And sometimes we know some of the disease-causing mechanism, but we cannot write the whole equation such that because of the disease, the mechanism, and then so the patient has certain type of phenotype. And then we have the therapeutic intervention. So we give a drug. We give drug to the patient, and at that time we have m to the nth power possibility. So we treated the patient when the patient had a diseased mechanism and such as getting sick, then we treat with a certain drug, and then the function would change. Okay? And again, we do not know how to write the equation explicitly. S prime, sometimes we know, sometimes we do not know. And then we cannot write these two equations. So we really don't know anything explicitly in a very precise form. Okay. So how do we deal with that? So we take a Taylor series expansion. The Taylor series expansion, that means a diseased patient and treated by a drug or combinatory drug, they expand as Fs prime plus the second constant, first order, second order, and the interaction terms plus high order terms of the drug. Okay? From all the experiment we have observed, the high order term is not really important. 10%, 5%, 20% errors. Okay? So we can just ignore these high order terms. And then we move this function to the left side of the equal sign. That's a key step. Okay, so one small step in mathematics, one giant leap for medicine. Okay, why is that? So we move, sorry. So we move this term to that term. So this minus this and equal to all this calibration constant and plus the concentration or the drug, first order, second order, and the drug combination. So what is the difference? The difference is efficacy. Is what we want to find out. Is efficacy or toxicity, efficacy minus toxicity. So that's what we find out if we put the drug and how does the patient respond. With this equation, we really don't have to know all the things we don't know. Okay? all we have to do, just we calibrate the patient with this constant, and then we know what the dosage and what kind of efficacy we're going to have. And it's a second order term. And when we move this to that side, and this is the black box we always talk about. Okay? So when we move to that term, and then it's indication agnostic. So that means any disease, we can use this equation. Okay? And also mechanism independent, because I don't have to know S prime. That's all. It's a Taylor Swift expansion. Move one term to another term. Then we have the very simple law to relate input and output. And that law works for all the disease, for cells, for animal, for human, for specific human. So PPM, phenotypic personalized medicine, basically become a calibration process of a biocomplex system. When we treat patient, we just measure input and output, we calibrate. After we calibrate, what is the best output you want, I tell you what is the best input you need. Okay? And also we know that this kind of biosystem response surface is not a linear system. Second order and the drug-drug interaction is very important. So the maximum tolerated dosage, additive maximum tolerated dosage doesn't work. Okay? And fortunately, doing that, most of the time the patient suffer a lot because it's a very high dosage. Okay? And then still, the efficacy is not very high. So why we go this direction become very easier, very easy. So one drug, parabola, I need 3 data points. Yes, we test the 3 data points and know what is the maximum. Usually, when you treat the patient, it's 3 to 4 drugs. So you need to calibrate 10 or 15 points. After 10 or 15 points, I know all the possible combination and what is the maximum efficacy and what is the most optimal drug dosage. So small number of these tests, and we calibrate the system, and based on this PPM response surface, and we got the optimal. We just did a TB test, TB drug development. We start with 14 drugs. If you take 14 drugs, each drug uses 5 dose levels, is 6 billion possibilities. There's no way I can test 6 billion data. But the 14 drugs, if you write the 14 drugs, that second order algebra equation has only 120 coefficients. So I do 120 coefficients, I will be find all the possible combination and what is the best drug and dose values for that, instead of a 6 billion data point. And the advantage of that is we can do that in a very short time. We can treat the patient before the patient disease is really getting serious. And also we can find the real optimum. Okay? So we can do that in a cell line. So in a cell line, we can sacrifice millions of cells. So we can, from a large number of a drug pool, I can narrow down a few good drug combinations. The dosage is not important for us because the dosage, when you go to animal, all changed because the metabolism and everything, the dosage we found that the best doesn't work that way. But that's the way which we are doing nowadays, use conventional approach. You find the best drug and the best dosage, they just scaling up. And of course, it will not work. Okay? So what do we do? When we go to the animal test, and we know with these 4 drugs are very good. So with a small number of groups and to test that animal, and then we can find the best dose for animal. After we get the best dose animal, shows it really works in a animal body. Then we'll go to human. When we go to human, we still drop out all the optimal dose test found out in the animal. We go to human, then the process will self-adjust, try to find out the best dose of that 4 drug in human. Okay, so far, we have 0 failure rate. Everyone, it works. All the disease, all the drug combination works very well. Personalized medicine. let's take one drug. Let's take one drug, and so that's the parabolic function. You have 3 constants. So we test the 3 constants, I can find the optimum. Okay? For patient 1, 3 drug test, we find the optimum. For patient 2, this parabola is not correct for that patient. So we do 3 tests, and we find the parabola for patient 2. And can get optimum. Patient 3, same thing. I test the 3 data points, 3 doses for that patient, and I can find the optimum for that patient. That's how simple the personalized medicine can work. Okay, so you may not believe how can use a parabolic equation try to treat a patient with such a complex system. And that's we did for kidney transplant, a retrospective test. Okay, so we work with a group of our kidney transplant unit on campus. UCLA has the largest organ transplant center in the country. Usually, for hospitals, they have 1 helicopter port to deliver organs. We have 2 of them. And my office is very close. So every day I was bothered by the helicopter noise, okay? And so what they do is the doctor uses these 3 drugs to control these 2 markers, okay? Tacrolimus and the creatinine. Creatinine is the mark to show the function of the kidney. And this one is to try to show how the immune suppression is good or not very good. And, but these 3 drugs have to keep these 2 markers within this shaded regime. Okay? How narrow is the shaded regime? For this one, it's about 9 to 12 nanogram per ml. 3 nanogram per ml of tacrolimus have to keep in that white region. If it doesn't, that's a day after transplant, and that's about 3 months afterwards. Like this patient just go up and down, and this the kidney function is not very good, so it's way above the limit. And after 3 months, and the patient most likely have to go through another kidney transplant. Okay? And it's hard for a doctor to control that, because usually the dosage is too high, and then drop it is too low, because the metabolism function is so complicated. There's no way the doctor can tell what is the right dosage. Too low, then go up, then just going oscillatory. Okay? If it is too high, and then the infection will take place. If this dosage is too high, the tacrolimus is a neuron toxin. So if you give too much neurotoxin, yes, you will not reject the kidney, but the problem is the patient will behave very erratically. Okay? The neural system has been affected, so cannot speak correctly and has very strange behavior. So what do? So first few days, the doctor said, okay, take this dose, give this dose. So we follow the dose. Follow the dose, then we also have the output. So use the input and the output of the doctor given, then we establish the second order algebraic equation. Okay? So after we set up the algebraic equation, we know the coefficient, then after we get that equation set right, then we tell the doctor, don't give the red drug, give the green drug. So follow all the green drug. That's how it works. Okay? So after we get the system calibrated, give the right dosage, and then you can get into the range and stay within that range to that patient. So these are the 3 patients. Drug A, drug B, drug C, drug A, drug B, drug C. You can see each patient that you have to give very different dose, okay, for all the drugs. And if we can do that, and you can see the markers can keep within the range. If you cannot do that, and the patient will not get the right dose and will not respond properly. So retrospective, the real doctors doesn't believe, say, oh, great, you just play the numbers. And then until about last year, we start work with liver transplant. Okay, so that's a prospective test. So it's really when the patient start to, after the transplant, and then we work on that and try to give the right dosage. That was very scary. First time someone's life is in my hand. And, so we start working on that. And that's a PPM patient. So that's our patient. That's a control patient. So that's a doctor's patient. Okay? And that's also, you can see the dates, and we can use these two drugs, and it can make it pretty good. Okay? And get into range very good. And sometimes it does not. And that's first time when it's dropped out was really scary. Because we know that's the first time going up, because now we don't know, and now it works right, and jump up. And because it works so well, and how can that happen? So we call up the doctor. The doctor said, yeah, this guy has an infection, so we give antibiotics. They don't tell us. They say, if anytime you change the drug, the equation changes, you have to tell us. And I even asked the doctor, say, if you give that antibiotics, whether it will really affect the tacrolimus reading. Some doctors say yes, some doctors say no, because they don't know. Okay? Because that's a routine protocol. They just do what they're supposed to do. And not only because of antibiotics. And if you give the tacrolimus in the emergency room, they go through IV, so go to the blood. Okay? And when the patient start recover, move out from emergency, go to the regular patient, and the people can start swallow and give tablets. Same dosage in the IV, and the response is completely different from if you give a tablet. So we have to recalibrate. We have to recalibrate consistently. Okay? So we have to do a moving window every few days to do that. And also, that's obvious, even without all the other change, because the patient, the body system change when you give the drug every day. So it either go better or getting worse, right? So the equation change, the constant change. So we have to do a continuous optimization. That's one of the control patient. Okay, the doctor give these two drugs and different dosage, and you can see first it drop very, very low. When it's very, very low, and then the doctor says, okay, I have to go up, but they have to take 11 days to reach to the limit. Okay? Because doctors based on titration. If too low, I give higher, and too high, I give lower, but doesn't know how much increment will give, such it will not go too high. Okay? Too low is infection, that's okay, antibiotics will take care. If it's too high, the patient will really get into serious trouble. Okay? So that's the first retrospective experiment we did. Okay, so this patient is really not doing very well. And so that's a prospective data which you just saw in the previous slides. So now after the patient treated, and then we said, okay, we use our method, can we do much better with retrospective test? So we did change the drug and the dose according to our parabolic response surface, and you'll see how much we can do. It's really surprising such a second-order parabolic response surface can handle specific patient, and doing so well. Okay? So life is pretty simple. Don't make it too complex. Okay, so that's another project we work with the Swiss group. Okay, so you can see, and you can see that all the surface is fairly smooth. And then we plot all the possible combinations. Okay? And the lower the better. You see all these possible combinations, only the last 2 or 3 is a good one. Okay, we know it's the best one because we know all the ranking list of all the drug and the dose combination. When we use a conventional method, we don't know how to do that, because if we do that, we'll kill thousands of mice. By doing that, we spend sequence about 100 or 75 mice. We can get all the combinations. We pick out the best one. Then we go from that point, we can go to human, or from that point, we can go to from the cell line, go to the animal test. That's why Nowadays, if you go from cell line to clinical test, the successful rate is so low. Sometimes you pick up suboptimal. From suboptimal, you say, yeah, it's pretty good. Then from that point, you go to cell line, go to animal to human. And the successful rate getting worse and worse. That's why it's getting very low successful rate. So the ability to be able to get all the ranking list of all the combination is critical to increase the successful rate for that personalized medicine, or even for ordinary treatment. So we are supported by Gates Foundation to work on TB. TB, most people, so TB already gone, it's not a problem. No, it's not true. Okay? TB is almost not a threat. It's a few years after Second World War. The antibiotics become available, so the drug are sensitive, and that's how most of the patients come. But now, the bacteria become drug resistant to the drug we're using. So 1/3 of the world population infected by TB, 1/3 of us all infected by TB, but it stays in a dormant state. And 9.5 million cases every year. So some people get activated, or some people get infected. And the death rate is 1.5 million deaths per year. Very large number, especially in Asia and in Africa. Okay? For drug-resistant TB, China has the largest number of drug-resistant TB in the world. So it's a very serious problem. But nobody had to, no pharmaceutical company tried to develop new drugs because there's no business in the States and in Europe. That's why Gates Foundation tried to provide research for us to do, or many places try to do that. So we take 14 drugs, okay, and try to do all the possible combination, all the possible drug, those combinations. And we come out with 2 combinations pretty good. So how much challenge you have to go through? 14 drugs with 5 doses each, 5 to 14 to the power 6 billion possible combinations. Okay? And if we do the regular way, we have to test one by one. No way we can do that. But if we use the parabolic response surface and only 120 calibration constant, that equation has 120 points. Calibrate that, we got all of them. Okay? So we got all of them, and then we first do a crude search, then we narrow down to 9 drugs, we narrow down to 6 drugs. And so we did all this, we always spend less than 1,000 in vitro tests instead of 6 billion. Then we get 2 very, very good combinations. There's nobody using 14 drugs. So usually in the clinic, use 3 or 4 drug combinations. If we start with 14 drugs, we have 1,365 combinations in 14 drug pools. We use 120-point tests. We know each of the best dosage for this 1,365 combination, and we know all the efficacy. Then we find out what are the best. You know how many are good? 10 to 20 out from the 1,365. That's good efficacy. That's why the successful rate is so low if you don't know the whole landscape. Okay, so we do have to know the whole entire drug dose landscape, and then we have much better chance to find out the good drug combination. So 10 out from 1,365, so much less than 1%. That number is very close to what the typical score nowadays in the pharmaceutical companies. Okay? Very small number percentage will be successful. Okay, so we did that from an in vitro experiment, and then we go to the in vivo experiment. So with the in vivo experiment, so we let the mice infected, so the bacteria load going up. At that point, we try to give the drug. So this group is a sham group, that's control group. So there's no drug given. And this brown curve is using the standard regime. This standard regime is It's started about 1985. So nowadays, a patient getting sick, go to the hospital, were treated by this standard regime. Okay, 1985, 30 years old. And then FSC regime 1, so it takes about 8 weeks and reach sterilization. That means no single bacteria in the lung of the mice. And the typical treatment, it takes about 24 weeks. Okay? And the FSC regime 2 only takes 3 weeks, go to 0 bacteria. And another thing is important, you stop the treatment and you should, the mouse or the patient should not have a relapse. Okay? And why? It's successful. We pick up 2 and it works, the 2 drug works well. It's because when we start to find this regimen 1 and regimen 2 and go to the animal, and we don't use the dose found out in the in vitro. Then we test about 10 parallel groups of mouse, each one with different dose and the drug. Then we've used that, re-establish a parabolic surface. Then we find out what is optimal dose for the animal. Okay? We also did the clinical test in Shanghai. From 16 patients out from 8 patients, and cured at the end of a 4 months treatment. The standard one takes about 6 months. So indeed, we can find we can optimize the drug dose from in vitro to in vivo and also to clinical tests. Okay. And what we have spent is about less than 2 years. And the Gates Foundation support to us on TB part is about $2 million. But for the last 10 to 15 years, just the Gates Foundation alone spent about $1.5 billion. Not a single drug ever been found out because it's all by trial and error. Now we know the whole landscape, and with a small number of tests, and we are able to do that. So this method, we work on that for about 7 or 8 years. We have applied to so many different type of indication. All that has been, the cell line test has been tested. The red one has animal test. And the one with that one, we either the liver transplant has been finished, and some of that is ongoing. And the multiple myeloma, we just started at Singapore. We started to recruit patients. That's people for the older people's blood cancer. And we use that for Chinese herbal medicine, work with Carl Zhen, Yi Kun, and Nancy Yip. And it also works for parasite control. Actually, once I went to Iowa State to give a talk. Iowa State is a farm state. They even don't have a medical school, but they have a lot of agricultural needs. So they said, why don't you try to use this way, use this drug combination, try to treat the patient, treat the parasite for the pigs. Same thing, they have 4 drugs developed 40 years ago. And it really doesn't work very well for treating the parasite for pig. And we use this way, we put 4 drugs in combination. We use about 20% of the dosage, and we can get a 95% kill. And now they use very high dosage, they get a 50% kill. But it's really a indication independent method, and also mechanism-free method. We can apply to almost all the cases we have. So how can we do that? I was an aerospace engineer, okay? And I don't know anything about biology. And the reason we can go to that is we really have to open our mind to work in the interdisciplinary area. Think about that. Nowadays, all the interesting part is at the interface of two or three or four different in the interface of the disciplines. when you put all together, try to work in the interdisciplinary area, it's art. It's not a technology. It's not engineering. It's not medicine. It's science. So how do you put all together, put it right? Just like how can you put the drug and the dose corrected, and you can get something very useful. Okay? And I always use in if you have the same ingredient, same thing to give a famous chef or give to me, even exactly the same thing, the chef much better than what I can do. Okay? That kind of art can be taught from one person to another person. He even doesn't know how that's come up. The liver transplant group we work with, the chief surgeon is the highest paid professor in whole UC system, $2.5 million. So I asked one of the surgeons, I said, does he really worth $2.5 million? Yes, he was $2.5 million. I said, why? He said, usually the liver transplant takes 6 to 8 hours. He can get it done 2 and 3 hours. Okay? And he cut here and suture here and cut there and get it done. Okay? And the most important, so whenever he got into trouble, he can get out. Okay? That's art. It's not just a technique. So same thing when you do the interdisciplinary, is how do you mix that in the right way at the right time, and you'll be successful. So most important is to think differently. We cannot follow the same approach. If we follow the same approach, even we try to improve a small step, and will not reach a really very, very exciting results. Okay, so our technique is really a top-down, goal-oriented technology platform. We talk about cells, we talk about animal, we talk about human. All these functions we do not know. Okay, and also it will be very hard to know. So what we do is we find a parabolic response surface. Just that simple. And we look into the output, we look at the input. After a few data points, we calibrate. After we calibrate, then what output you want, I can tell you what the input you need. That's all. So at the very first slide, we talk about the challenges. So now we are able to use a small number of, so now we are able to identify a very small number of optimal combinations out from 1,365 combinations. Okay? Then we can narrow down, and then we can proceed from cell line to animal to human. Okay? Because we recalibrate, reestablish in each of the different biological systems. So that's the phenotypic personalized medicine. So these are all publication lists. One thing I really wanted to bring your attention. This morning I spent half hour with Henry because we have a golden conference, and the right happens in this building. Okay, and that's the first personalized medicine golden conference. We invite 3 groups of people. One is phenotypic personalized medicine. Another group is genotypic precision medicine. And all of that need a lot of advanced science, advanced technology. So we get really top speakers from these 3 groups. And it will take place in July 10th to 15th in this building. And that time doesn't have a hotel. Now we have a hotel. That's great. Okay? And so hope all of you or some of you will be interested to participate in this activity. And I think everybody will learn quite a lot, and we'll see. Most important is the first conference on personalized medicine, so we can lay out our future, our direction in this conference. Thank you.
Very good motivational content, and also it's kind of like you said, it's interesting to see seemingly human body can respond quite simply. Right? But on the other hand, also the approach is very, I would say, very simple, and in some cases, some sense, actually rudimentary. Because parabolic mapping, the response survey is well known. There are a lot of papers on this. Whether I think there are many ways to refine it. That may or may not be the most critical to get where you are. I certainly appreciate the approach, which is very effective. However, I think the obvious question is variable to variable kind of interaction. And a problem may or may not be the most effective way to represent in terms of response surface. There are many different treatments in this regard. You can use kriging, you can use neural net, you can use even Markov chain. So these are actually in aerospace, as you know, many of these methods all have been tried, and because degree freedoms are too many, it's very hard to treat them. Here, you have much fewer in that sense, even though the data set can be large.
The field is very important. That's a time quick.
That's right.
Okay.
Yes.
Because
So,
you don't have time to play.
yeah.
Okay. You
But
get it right.
It's more than that. I think it's because the number of variables are still much more manageable. Okay.
Yeah.
So I guess my question is, first of all, to go to larger data set, I think there is a lot of room for some very straightforward kind of model testing in terms of the fidelity, in terms of the correlation, and
That's all one year's
and
job,
also in
not my
terms of
job.
in terms of design of the, more importantly, in terms of design of the experiment. Because right now, I think oftentimes not the best way to treat it with certain kind of interval, which is a structure.
Yes, so we say 3 drugs, we need 10 points, okay? So that 10 points, and you need to use, you can use the experimental design to find what are the 10 best points. However, Wen Yang told me, he said, no, you do random selection, will be even better.
Yeah. No, it's not random, actually. There is some semi-random selection. Random is not the best, I'm sorry to say.
not entirely random. Actually.
Yeah, exactly. So there are some very well-established randomized design experiment approach.
Yeah.
I think these there are some obvious way to further improve the methodology.
Yes, that's a very important point, okay? But another even more critical criteria is still the time, okay? You can improve, maybe can improve better, but you don't have the time to do that. So you have to find the shortest number of calibration point to get it done right.
They are not contradicting.
Yeah, sure.
Okay, the time and how to design, they can be reconciled. But it's very interesting work.
Yes. Wow, the biologist.
Yeah, biologists
always want to kill me because,
always
because I don't need to know FS.
So Chih-Ming, it's a beautiful talk. I have one thought, whether it makes sense. The other is a question. The thought is that, is it possible your methods can very elegantly work in the living system because the living system by itself, it's a converging system.
Yes, that's right.
Right.
Okay.
Because you cannot deviate certain parameters.
Right.
Parameters can be black box. It doesn't matter. Once you deviate to a certain level, either system bursts, or you just drive them back.
Why it doesn't deviate? It's okay. This actually, if you look at a high level, is a complex system. Okay, the turbines are past life. Okay, the biosystem, or the financial system, are all complex systems. Complex systems, 4 fundamental characteristics. First one, there's a large interacting elements. Okay? Second one, actually that one make our life is tough, is self-organized into a complex system. Then they have emerging properties. And when the emerging properties come up, you cannot go back to interpret why this emerging come out is from a limited subset of the building blocks. That means the inverse property become very difficult. Fourth one, that's when you talk about that save our life because it works. Fourth one is after long evolution, the whole system is very robust and resilient. Very robust and adaptable. Okay? So that means if you give a perturbation, the response has to be smooth. You will not die. And so smooth is second order system. So because of such a millions of life evolution, all the one which is not adaptive robust died already, doesn't exist nowadays.
So related question is that, so with this method, if you pick up many of the potential lead compound failed in different stages of clinical test, you would potentially...
Everybody try to make money by doing that. And you remember the slides I showed after 3 single molecules, which it doesn't work, I put the 3 together right and then become very effective. Okay? So there's a possibility. Not all the drug like
Yeah.
So that's why a lot of time when you say, okay, that is failed, the failed is not because the molecule is not right, its dosage was not right. Okay? And so that's why we can be successful. We are successful is because whenever we go to animal, go to human, we recalibrate, readjust the doses.
Yeah. So it's beautiful, but a practical issue for human patients is that how you implement it. In other words, is that for going down to the individual clinics, individual patients, who will be doing the calibrations and how to do calibrate? Will this kind of implementation be a rate limiting step in the real clinical settings?
Okay, and that means a scale up. Okay, like the liver transplant. In the morning, the doctor tells us, hey, the drug you give today, what is the marker value? Okay, so in 4 hours, and we have to find out what the dosage you give today. The next day we check that out. Okay, so for this first time is 4 patients and myself and the postdoc and we or graduate student work together. And first 2 weeks was pretty nervous because we don't know what we calibrate and we've tried out, it works, and sometimes extra drug was given, and then the patient move out from emergency room into the regular patient room. A lot of variables. So after a few weeks, then all I do is just get data and use make a parabolic surface and give this drug. And another difficulty is for this liver transplant, first is 3 drug control. And when we really start, and the doctor said, no, you can only control 1 drug. Because in the past, they don't know how to control 1 drug. So they make it 2 drugs. They have from 30 years experience. They tell precisely what do you change. Okay? And so what does that tell us? You have multiple variables, so we have only 1 equation. Okay? That's why if we can have 2 or 3 drugs, actually we found if we have 2 drugs, then we can do a simple programming and tell you what is next drug for treatment. So it's a very tolerant system because it's a complex system. Yes?
Chih-Ming, I have another biology question. So when we talk
people
about...
will kill me.
Okay.
One is biologist, another is the doctors. Okay, when we do the experiment, I said, hey, after I finish that, all you guys lose job.
Well, it's not really a biology question.
The surgeon said, no, you cannot replace my hand, or the other doctor will lose job. Okay, anyway.
My question is, when I look at your the FSC, the input and output, so we have input that we already know what the output should be. Because when we look at the output for the liver transplant, you look at the marker that related to liver transplant. For the HIV, we look for the infection rate for the HIV. But when I think about the problem or the disease of individual, so you already mentioned, sometimes diabetic always come out with the cardiovascular function problem, always come out with the ulcer problem, with the stomach problems. So in that sense, have you ever tried the FSC, for example? You input that at a different methodology and the output they are different. For example, you look for the output that for the marker for immune response. But at the same time, the patient have the cardiovascular function problem. Have you looked at the NO productions? So in that sense, that will make your pictures more complicated, is more bigger. So I want to hear your comment.
Okay, basically you said, okay, if some patient has a disease A.
Yes.
Okay, and then also he has disease B.
Yes.
and you want to try to fix both disease A and B.
And that is normally happen.
Yes, normally happen. Actually, most of the serious death is caused by disease B, not disease A. Okay? Like a lot of the HIV patients died from TB.
Yes.
Okay? And yes, so that means that the output, originally, we just talked about one drug, one output. Okay? And now you have two output. I have to treat both patients with both disease at the same time. Okay. Then in that time is we have to find out the correct drug dose, drug combination. So the drug should be, some drug can treat A and some can treat B. Okay. Then we try to go in through the test. And by that time, it's not 3 drugs, maybe have to 5 drugs. So number of tests become longer, and time need longer. And whether we can really do that, and we have to test that out.
But in principle, that could be
Principle
doable,
can be
right?
done. In principle can be done. And also depends on how serious is the patient. Actually, you'll find out that whenever anyone getting sick, go see the doctor right away. If you see it right away, it's much easier to handle. That we can see that when we treat the HIV patient. Okay? HIV patient, if viral load is very low, and then we can do it right away. Doesn't need that much time to treat. And when the viral is very high, and then there's a problem. 16 of the TB patient we can get it right, and 2 doesn't get it right because 2 of that has the highest score of disease. Okay? So it's good. The drug treated very well to the end of 4th month, but not complete cure. So then we take a longer than 4 months to treat. It's a lot of parameters in there, and then you have to select the most critical one, try to handle that.
Thank you for your excellent talk. I think this is a really revolution in medicine. And my question is, then can we really use this kind of a micro nanofluid with integrated sensor, biosensor to monitor this kind of a patient, or so that we can do this kind of real-time calibration?
Yes or no? The reason no, okay? All the tests we do have to use a traditional FDA-approved test method, okay? And because that test result has been tested and also accepted by the community and also has legal status. Okay, talk about a microfluidic sensor, so far, there's no single microfluidic sensor has ever been FDA approved. Okay, has ever been approved. And actually, Vincent's first sensor will be FDA approved maybe 6 months from now. That's for urine test. There's no way you can get a blood test go through FDA at this moment. It's legal, and there's both is a regulation issue.
I'm sure we have more. I have more questions, but we can wait. Let's thank the speaker. Okay.